Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're happy you made time to dial in today. We will be talking about how AI helps advanced plate-based screening and imaging technologies for high-content screening with Peter Skewes-Cox today. Good morning, Peter.
Good
Glad to
morning,
have you.
Dennis. Thanks for having me.
Yeah, absolutely. It's a pleasure to have you here today. So Peter studied molecular and cell biology as an undergrad at UC Berkeley and bioinformatics as a PhD candidate at UC San Francisco. His graduate research centered around developing computational tools to detect new and divergent viruses for infections of unknown cause. In 2011, he joined the infectious disease department at the Novartis Institutes for Biomedical Research in Emeryville, where we met actually, and he was following one of his mentors from UCSF. Over the past 14 years, his work has evolved from genomics-based antiviral discovery to leading teams in computational biology and AI-driven imaging analysis, spanning the entire Novartis pipeline to identify drug targets and also chemical compounds that modulate them. So welcome today, Peter. We're honored to have you here.
Oh, thank you so much, Dennis. Happy to be here.
Awesome. And yeah, in the good old Drug Hunter fashion, we wanted to start with a few questions here to get to know our speaker a little bit better. So Peter, can you talk a little bit about what inspired you to start a career in drug discovery?
Yeah, happy to. So as a child growing up in the Bay Area, I was always kind of a computer geek and a math nerd. But when I was a teenager, I got extremely sick with severe aplastic anemia and ended up having a life-saving bone marrow transplant. And since that moment, when I was an early teenager, my life sort of changed. So that's the moment where I got into science, started getting interested in biology and studying immunology as an undergraduate, really trying to understand what had happened to me. And from there, with the opportunity to come to Novartis, I really wanted to take that opportunity to help other patients in the future. So that's sort of how I ended up where I am today.
That's an amazingly inspiring story. And yeah, for the audience, I didn't know this about Peter, although we worked together. So I think that's an extremely inspiring story. And yeah, there's no better motivation than that, I think. So yeah,
Yeah.
thank you, Peter. Very, very inspiring. What's your favorite molecule, Peter, and why?
So I am not a chemist, as you know very well, but I would have to say, and maybe I shouldn't be choosing a competitor compound, but I would have to say sofosbuvir was my favorite compound. Not because I have some emotional attachment to the chemistry, but because I learned so much about drug discovery when that molecule came out. At the time, Novartis was investing heavily in hepatitis C virus when we were in infectious diseases, and we were looking at different targets and different drugs. And when sofosbuvir came out, it was a bit wild because Gilead had pushed billions of dollars into a compound, and I just had trouble wrapping my mind around that risk. But it ended up paying big dividends, and it basically cured hepatitis C. And you hear things growing up, oh, the pharma companies aren't looking for the cure, man, or whatever it is. They did it. They made a huge strategic bet and they cured that virus. So I think that would probably be my favorite molecule, just in terms of like bringing everything home and contextualizing the importance of our work.
Yeah, I love that. That's a really an amazing story. And yeah, I love the motto, last in class, right? That
Right,
really solved the disease. If you're last in
right.
class, there's nothing else you need. You're last in class. So that's a motto, I guess, in the infectious disease space. So yeah, really cool, inspiring story. What are you most excited about in drug discovery today?
Well, you're going to hear about it shortly, but it's
Oh yeah.
really trying to bring some of the AI developments that are going on across industry and applying them to drug discovery for actual impact. There's a lot of hype around AI these days. There's a lot of problems and concerns folks have with how it's being applied and the effects it has on society. What really excites me is distilling what's good about it and its powers and applying it for good uses. So I hope today I can convince you that there is some promise that's worth following up on applying these to drug discovery.
Amazing. Yeah, can't wait to hear about it. And yeah, maybe can you tell us a little bit more, if you could go back to to the beginning of your career, what advice would you give your younger self? You've been in drug discovery for over a decade now. You've seen so many different programs across the Novartis portfolio. What are things you wish you knew earlier in your career?
Hmm. Hmm. I would say there's two domains of science and technology that I wish I had focused on more as a student. The first is statistics. I only took a couple stats classes and they were sort of introductory. I wished I had done more as an undergraduate there. The other one is to pay more attention in organic chemistry class. I didn't know it at the time when I was sort of dozing off and couldn't wrap my mind around the synthesis problems, but I didn't really like chemistry. And now I've come all the way full circle to realizing, wow, this is super cool. And I loved hearing talks from chemists like yourself and like Matt and other folks that we've worked together with over the years. It's taught me so much. But yeah, if I'm not kicking myself about once a month thinking, oh, what was that about again? So those are the two things, statistics and chemistry. I wish I had focused more on that in my earlier studies.
I think that's good advice. Every chemist or many chemists, I guess, will tell you they wish they had focused a little bit more on biology and learned more about biology. So I think this is kind of a mutual feeling.
Yeah.
Yeah, so that's great. Yeah, Peter, last question. What do you most like about Drug Hunter?
I think the thing I like most about Drug Hunter, and this is actually sort of where this conversation comes from today, is I ran into you in Cambridge about 6 months ago, and I saw the other dentist give a talk. And really, I think the thing that was cool about his talk was the storytelling behind each of the molecules. As I've mentioned a couple times now, I'm not a chemist. I don't have that intuition about the chemistry itself, but to see the full stories of how these drugs come to be from the very early preclinical work to actually helping patients. That aspect of Drug Hunter, reading the blurbs on the website, hearing Dennis talk about the top 10 molecules of 2024, that I think was the coolest part for me because it really contextualizes what we do in the broader pharma context.
Awesome. Yeah, amazing. Thank you for that nice compliment. Love to hear that. Yeah, before we dive into Peter's presentation, I want to briefly show folks the platform and show very briefly again one thing that you can do with it. So we have our full text search here. You can kind of go right here on the top right. For example, if you're interested in compounds that were discovered with high content screening, Peter's going to talk about high content screening in a phenotypic way today. And if you're interested in which programs was this like pivotal and which programs was this leveraged, you can really easily type this in the search here and immediately find projects where this was pivotal. So let's click into this Novartis case study here about a gut-restricted soft drug to treat cryptosporidiosis. So here you can, in our case studies, you can really easily see some of the highlights here. What makes this program special? Why is this a program you should pay attention to? In the introduction, you get a lot of context around this particular program, like what other compounds are used in this particular indications. What problems is this program solving? You get a lot of background here on the target and how it relates to the disease. And then really the full discovery story, including, again, what kind of screening method, what led to the initial hit on the program, and then how was this program really run from hit to lead throughout the lead optimization? What were some of the challenges that were solved on this program, and how were they structurally overcome? What kind of techniques did the team use to overcome all those challenges? And even oftentimes, we talk about structure, we talk about selected properties of the molecule. If data is available, we'll talk about clinical data as well as the synthesis here for you to get some inspiration of how you can quickly put those molecules together. And yeah, you can find many more. We have approximately 700 of those case studies available online today. And with that, I will stop sharing and hand it off to Peter for his talk about AI meets plate-based imaging, harnessing hallucinations for high content screening. Really excited about this talk, Peter.
Thanks so much, Dennis. All right, let's roll then. But first, I just want to acknowledge that I basically have done very little of this work, and the work has happened in our teams in data science in Novartis. I want to specifically acknowledge Minhua Chu, who leads our imaging and deep learning group from down in San Diego, Steve Cook, who has worked on this profile diffusion model that I'll talk about, Jian Fang, who has worked on the in-silico labeling problem that I will describe shortly, and William Godinez, who leads Steve and Jian, whom I've worked with for many years now. And so, yeah, and of course, all the collaborators in data science and really our experimental collaborators who generate all the data and feed these models for us. So just thanks to them. Also, thank you, Dennis, for having me. It's really good to be here. Thanks to Laurel for keeping me organized. And without further ado, I will get started on the talk. Here's a brief outline of what I'll talk about today. I want to give an introduction to AI and high content screening and really where those two things meet. Hopefully there's no screaming. I also want to tell you a little bit about how external innovation inspires our approach to internal challenges. As we've discussed, our role here is to accelerate drug discovery and get molecules to patients as quickly as possible. But we also need to stay on the state of the art. So I want to talk about that balance a little bit and our approach to doing so. And then for the meat of the talk, I'll talk about some generative AI methods and how we're applying that to high content screening, hopefully replacing some of the more laborious or expensive experimental portions of that workflow with AI methods. And then I'll give you some thoughts with a look towards the future at the end. Okay, so first, let's get some definitions straight. We hear AI, machine learning, deep learning. What do we really mean by these things? Well, it turns out when we talk about AI, we're talking about the superset of all methods that mimic human intelligence in some way. And that can be very narrow. It doesn't mean you have to have a sophisticated model. It could be as simple as a text box that you enter your name in and it says, "Hello, Peter." Right? That's mimicking human intelligence in a very narrow use case. A subset of AI is machine learning, and that's where you're giving a model examples of data, and the algorithms are able to learn from that data. And deep learning is a subset of machine learning where that process is done with multi-layer neural networks, okay? So when we say AI, we're talking about a number of different things, but here's some examples. So AI could be something as simple as a tic-tac-toe solver, and you can tell that I made that image with generative AI because it's got 6 X's and 3 O's, thereby breaking the rules of tic-tac-toe. We could have a spam filter. That would be an example of machine learning where perhaps the content of an email and the sender are the pattern that you train on to sort of triage your incoming emails, whether or not it's spam. But for a problem like protein structure prediction, that's usually done these days with deep learning. For the sake of this talk, I'm actually just going to be referring to all of this as AI, but actually when I say AI, I'm typically going to be referring to deep learning. And I'll show you this tweet that I liked from Matt Veloso, who I think he's a VP at Meta. This has always resonated with me. He says, if it's written in Python, it's probably machine learning. If it's written in PowerPoint, it's probably AI. So as the house style, I'm in PowerPoint right now. I will refer to it as AI, even though it's probably machine learning. Okay, so deep learning, multi-layer neural networks. What the heck? All right, well, we know what neurons are in the biological sense. Neurons have dendrites, which can receive signals from adjacent cells. Those signals are integrated in the cell body or soma. If the signal accumulates and exceeds a certain threshold, that neuron will fire that action potential down the axon into the axon terminals to adjacent neurons. Okay, so a neuron itself is simple. I mean, as simple as a biological system comes. But the human brain, you've got hundreds of trillions of connections between like 100 billion neurons. And you get that complexity from the integration of these simpler units. So in an artificial neural network, we're basically doing the same. We're mimicking that human biology. We've got inputs that come in from a previous neuron, and that's just basically a vector of numbers. You've got weights to say how important each of those inputs is to the particular neuron, and those are integrated together just like they are in a cell body in a biological neuron. In this case, you multiply the weight by the input and you add them all up, and it's adjusted by a bias term, which you can think of as the threshold that you need to exceed to fire. So it's basically some simple math that's passed to an activation function, which may say it may be a sigmoid, it may be some other shape, and that determines whether or not this artificial neuron fires. So by connecting these simple mathematical units together, we can actually really start to model complex data from these very simple mathematical functions. So the math, like multiplication and addition and some vector math, that's been around for hundreds of years, right? What's changed now? Why is this suddenly so important? Well, I would argue that when AI is less hype and is truly disruptive, it's when you have this confluence of three things. It's when the algorithms and the models themselves, the hardware used to process them, and the data that you have that you're inputting all sort of align at the right place in the right time. And so I have a really key example here that sort of is thought of as like the paper that kicked off the entire deep learning revolution. It's from 2012, which in AI years is like a couple millennia ago. But you can see these authors here. You may recognize some of them. Krzyzewski, he went on to Google, worked on Waymo, the self-driving cars. Ilya, he started OpenAI. He left due to ethical concerns. And Geoffrey Hinton, who won the Nobel Prize last year for his role in artificial neural networks, in large part because of this paper right here. So at the time, it was just a small paper that was presented at NIPS, the conference. And what was cool about it at the time was it wasn't really taking any new ideas per se. Convolutional neural networks, the model they were using, that idea had been around for a while. Alex Krzyzewski was playing with graphical processing units. So these are the GPUs that are used to do like 3D rendering on your computer. At the time, the biggest ones were used for like video games, shoot 'em up games, and they were doing focusing on a task called image labeling. So what's in the picture? Think about Google Images. You can pull up pictures of cats, tigers, fruit trees, whatever it may be. But where this all really came together at the perfect place and time was that Ilya's ideas about making these neural networks deeper with more neurons, more complicated, was met with Alex's desire to put some of the programming on these graphical processing units. Turns out that the math that's being done to do 3D polygon rendering and visualization is a lot of the same math that I showed before in processing artificial neural networks. So he was able to accelerate this task by putting it on GPUs. And because we, at that point in time, had a lot of digital images, right? When we were kids, it was just photographs. But now that we have so many digital images, many of them with labels, they were able to actually make a real advance in the accuracy. And that sort of kicked off this whole revolution. Okay, that's the AI piece. On the high content screening piece, high content screening is a key tech for drug discovery. We've got these imagers. We're able to put cells on plates, take pictures of them, capture rich morphology, extract different features, and we have downstream drug discovery use cases, whether it's hit finding or looking at mechanism of action, prioritizing triaging hits, looking for safety signals, et cetera. Okay, so this has been key, and the instrumentation is advanced, and we're generating more and more data that's more and more complex as we are getting more advanced cellular models too. But AI has really revolutionized cellular imaging analysis because of the way that we set up these experiments. So we're able to take a cellular system and perturb it in some way. Maybe it's with an siRNA, maybe it's with a compound. Get these phenotypic images, and the phenotypic changes that you see are indicative of what the drug or perturbation is doing, right? So the conventional pipeline would be you sort of draw boxes around the cells and extract some descriptors, maybe cell size, the ellipticity, the granularity, sort of human features. And then you're able to do some downstream machine learning and analysis on it. Where deep learning has really disrupted this is you're able to just go straight from the images to the downstream analysis. You're asking the neural networks to pull out those features that are important for driving a particular phenotype. And this has really changed the game for us. So how do we actually apply this in Novartis? It turns out that staying on the front lines of AI really necessitates a new way of working. I mentioned the GPUs themselves, the hardware. We got to have the latest hardware to be able to use the latest models. We also need to be generating actionable data. We need to have rich data sets that are really probing the biology of interest. Another key step is that we're always monitoring the literature and what else is out there for inspiration. So, some of the funding decisions that have happened recently for academia are a little bit troubling for us because we worry that some of the basic research may not be producing as many of these new models. But definitely want to say thank you to those of you who are in industry who continue to publish. I think the onus is on us more than ever to continue doing so and sharing our ideas to the best of our abilities so that we can continue to work collectively here. And the last thing we need to do is to have a system for quickly integrating and building and testing these new models. If we have to start from scratch each time a new model comes out, we're never going to be able to get up and running because I mentioned we have to always be impacting drug discovery projects. So I want to give a couple examples of this before I jump into the generative piece. So the first time we really did internalized and external innovation and applied it in high content screening was actually shortly after the paper that I mentioned earlier, the ImageNet paper, or the AlexNet paper rather. I mentioned that they're able to build a deep convolutional neural network and label images. This has got a dog and a cherry. Well, it's labeling it Dalmatian. We can get sort of probabilities for each of these labels. It did pretty well. Well, William Godinez, whom I mentioned earlier, was inspired by this paper and thought we should be applying this to microscopy images. And what he did was take an older microscopic analysis approach called multiscale, and that breaks the images down into the whole image and then smaller and smaller subfields. And he built a convolutional neural network that took each of those zoom levels as input. And what he was able to do was actually label, instead of this is a dog, this is a mushroom, this is a car, he was actually able to label the phenotype with high accuracy, without any segmentation of features or anything, just the images, going from the images to the labels, thereby taking what we had seen published and applying it for impact internally. I've got a number of references listed here that you can check out if you're interested in digging deeper into them. Another example where we were inspired by what's going on internally is you may have seen some of these. It's a little bit creepy to me, but there's like these surveillance models that are able to track people through crowds on video. Well, we were encountered with a problem where we needed to track a cellular organelle in images. And so we actually took some of those same models and applied them to track these organelles at the same time, where motility was a key readout here. You can imagine that this is much like tracking the faces in the crowd. So this is another example of us being inspired by what's already out there and applying it internally to a high content screening problem. What can happen though in crowds is people can walk in front of each other. And this is also a problem in autonomous driving, where you have to be able to distinguish between a human and a car. Or if someone's walking behind a car and you're taking video and your car is driving itself, you've got to know that that's a human for safety reasons. So we were inspired by that because sometimes we get these occlusions in cellular images. So if we have an image of organoids growing and we want to know the size of the organoids and that's our readout, we need to be able to distinguish these overlapping things. So we took some of the same models. One's called YOLO, you only look once, which allows us to go through an image just once and have the sort of disambiguated segmentation rather than having to go through once and then rescan it again. These files are big. These models are expensive to run. The quicker you can do it, the better. The more we can deliver impact on a project. And so what that lets us do is actually get a more accurate measurement despite a potential partial occlusion. And in fact, since then, we've taken this to the next level because some of this actually happens in 3D and we'll have Z-stacks. You can imagine one organoid is on top of another. Turns out we can even do 3D segmentation to get volumetric measurements of these things, which has been super cool. Again, inspired by published methods. Okay, so that's sort of where I'll leave it on the how we're putting external innovation to use on our internal projects. But I want to talk about sort of the next level, which is really inspired by generative methods. So I mentioned the sort of rough workflow for high content screening, I keep saying it, is that we apply a perturbogen to cells and we stain those cells and then we take a microscopic image. There was a paper published around 7 years ago now that did the actual staining in silico. So that enables you to take a brightfield image and actually get a stained image entirely via AI. And this can be a real accelerant, as I'll show you. Turns out these models are inspired by the colorizing of black and white images. So you may have seen these old images in black and white that folks are able to colorize and put on the internet. Those same models are what we're using actually to do this. Of course, it's trivial to take a color image and turn it into black and white and use that as your training set where you have sort of paired images, and then you can show it a new black and white image and it'll show you what it would look like in color. But for cells, it's not as simple. So actually what we do to train these models is we take a bright field image and we take fluorescent images. So in this case, I've shown a few different stains here, and we build a model for each one. So when you see this bright field or black and white image, produce this stained image. When you see this bright field image, produce this stained image, etc. Then what we're able to do with those models trained and validated is we're able to apply them prospectively to bright field images. We no longer need to take the fluorescent image, right? So we can get these merged outputs as if they were, just actually stained. And this has a number of advantages. One of the advantages, of course, is that it's cheaper and faster. Bright field imaging is just not as expensive. It's fewer reagents, right? And it goes much more quickly. There's no laser excitation and all that. Another one is that you can actually multiplex infinitely. There's typically physical limits on these microscopes as to how many stains you can image in one run. And in this case, doesn't matter, right? We have the bright field image and we can stain to our heart's content. Another one that's key, and maybe I didn't appreciate it at first, is that with staining, you tend to get some observer effects, if you will, right? The mere act of staining could be seen as a perturbagen. So if you're just doing bright field, you're actually sort of hands off here. Some of these stains are toxic or mildly toxic. So we can do things like time lapse now, where we're monitoring over time that we couldn't do before. And finally, we have some anecdotes. I won't say this is like for sure, but sometimes you can get like little smudges on fluorescent images. The model doesn't produce those smudges. You don't always have those artifacts. So I won't say that it's always cleaner because it does miss some things, but that is kind of an interesting thing that you don't see those smudges that you might see. So we have to verify, of course, that these models are working. And the standard way that we do so off the bat is to basically look at correlation of pixel intensity between the actual image and the in silico image. That's not always perfect, but really it's not a question of like if it's perfect. There's the classic quote like, all models are wrong, but some are useful. So keep that in mind as we go through this, right? We really want to assess like are downstream tasks just as good? Because obviously there are benefits in terms of speed and time by taking this approach. So one of the things that we tested downstream was phenotypic hit expansion. So for the chemists on the call, you know that if you get hits in a biochemical screen, you can do a hit expansion and look for different analogs and pull those in and test those too. But it gets a little bit more complicated when you're doing a phenotypic hit expansion. So we set up this experiment where we took 50,000 compounds and generated 10 different in silico labeling models. So 10 different stains. And for each of those, we performed a downstream image analysis and we captured what we call the ISL fingerprint. And we sort of put that in a database. And what that enables us to do then is to take a query molecule for which we have an image and get its in silico labeled fingerprint and compare it to these ones here and sort of pull in nearest neighbors and basically do that similarity search in a phenotypic space rather than doing it in chemical space. So we sort of put this to the test here. And what we did was we tracked down, we had about 300 assays where we had a good overlap in the compounds that were running them. These are both biochemical and phenotypic assays. And where we also had run them in this in silico labeling training set where we had actual images and in silico labeling images. So the first thing we did for each of these assays is take 10 random hits. Now the in silico labeling was done in U2OS cells, so there's no guarantee that the hit from this assay is active in U2OS. So we do a little triage to say, is it actually active? Is it different from DMSO in the imaging assay. And we pull in the 50 nearest neighbors. And we basically ask the question, like, are those hits when we pull them in? Are they hits in the assay too? If we look at the ground truth, and we do this with real images, we see that for those compounds that are active in U2OS, in other words, those that do show some sort of phenotype different from DMSO, that we are able to enrich for hits about threefold if we compare to the initial hit rate of the assay. In other words, this expansion does seem to be working. We are pulling in hits better than random. If you look at those that are not active, sort of negatives, we see that we're not really enriching, which we would expect, right? So it turns out we're not only picking positives better, but we're also picking negatives just as well. That's when we do it with images. Now, if we compare that to the in-silico labeling approach, where instead of using the real images, we're just using the fingerprints from the in-silico labeled images, turns out we see almost the exact same thing, a threefold enrichment here. So that really gave us a lot of confidence that even if the model is not perfect, we're still able to do phenotypic hit expansion with these. And so that's actually given us confidence to run the entire Novartis deck in brightfield. So that's going on right now. So we will have in-silico labeling fingerprints for 2 million compounds times 10 different stains in a database for phenotypic hit expansion. I'll just add that if you look at the actual assays, some of them are biochemical assays and some of them are phenotypic assays. We do much better in the phenotypic assays in terms of enriching, and that sort of makes sense, right? Okay, and of course we've compared to a baseline here of just the chemical fingerprints, which don't perform as well, and you can basically see indications of activity cliffs. Okay. So that is one example of how we're using ISL to do phenotypic hit expansion. Maybe if you come back in a year, there'll be a paper about this. But we're applying this to projects now, which is pretty exciting. The last thing that I would like to talk about is a little bit sci-fi. I'm going to warn you. But I think it's pretty cool, and I'm really excited to talk about it. And that's this pDIFF. We got a little bolder with how we might accelerate drug discovery with AI, and said, what if instead of getting rid of staining, we could get rid of the entire microscopy piece? What if we could just go from the compound and get an image out? That might sound ridiculous, and frankly, even saying it out loud, that sounds ridiculous to me. But we do have examples of this that we're inspired by in the field. So you may have heard of stable diffusion or other generative imaging models. You may have seen some generative imaging in this talk already. But you can say things like, show me a photograph of an astronaut riding a horse. That's not in any training set of real photographs, right? I don't think that's ever happened. Maybe there's some conspiracy someone can fill me in on. But you can get what looks like a realistic photo of an astronaut riding a horse, right? Well, we see in papers all the time, like, okay, here's an image of U2OS cells treated by a checkpoint inhibitor. What if instead of having to take that image, we could just give that description? I want an image of U2OS cells treated with a checkpoint inhibitor, and we would get this. Could a generative model hallucinate a cellular image? Yes, that's definitely the answer. Is that useful at all? Is it accurate enough? I think that's another question that I hope to convince you is yes also. So this approach is pDIFF. It's profile conditioned stable diffusion model for high content imaging. Profile conditioned. So what does that mean? Well, on the previous slide, I showed you where we had an image of an astronaut riding a horse. That image generation process is conditioned on that text prompt. We are saying astronaut riding horse. That's what you're passing to the model. So it knows what image it's going to generate. Otherwise, it's just going to generate random images that look real, but will have no sort of, you're not pushing it into a particular corner. So we're using that same approach, but instead of having the prompt be, show me U2OS cells perturbed by something, we're not passing a sentence of text. We're actually passing what we call an in silico bioactivity profile. I don't have time to get into what pQSAR is and how that works, but suffice it to say that basically for any Novartis compound, we've got a machine learning method that allows us to predict that compound's activity in all of our assays. I think we're over like 14,000 assays now. And so you can sort of view that as a fingerprint. What's the compound's activity, predicted activity in this assay versus this assay versus this assay versus this assay. And it turns out when you pass in a sentence to one of these generative models, what also happens first is that sentence gets vectorized. It gets turned into a string of numbers. So we're basically doing that same thing, but instead of embedding some semantic meaning, we're embedding some biological activity meaning, okay? So that's the key thing that we're doing here. And then we're using a standard image generation model, and we're generating a microscopy image, okay? So we have a bunch of examples of known drugs where we have their predictive bioactivity and we have their images, and we're training a model like that. So what we're saying is that this enables a purely virtual phenotypic profiling of novel chemical matter. So how do these stable diffusion models work? And you'll notice the GIF, hopefully it's rendering properly on the stream right now, where we're actually running pDIFF to generate a cellular image. So that'll loop through while I'm talking right now. But the training of one of these stable diffusion models starts with a desired image. So it could be someone's face, it could be a picture of a dog, it could be a picture of some cells. And what this model does is you add Gaussian noise to the image. So you blur it basically. And you ask the model to train to predict what noise was added to go from this image to this image, okay? And you do that repeatedly. And so what the model learns is how to denoise, basically. So that's the training step, moving from right to left. When you actually go from left to right and you provide it a prompt, it goes through the denoising process. So you're saying, I want a picture of a man's face, and it knows how to predict from noise what a slightly denoised image would look like, a process that's iterated repeatedly. And so the way that I think about it, and the way that Steve Cook, who pioneered this work, describes it, is sort of like building a sculpture. You've got a big block of marble, and you're kind of chipping away at what's in there at each step. And eventually you have this refined image at the end. Okay, so this looks to my untrained eye like a cellular image, but it's probably wrong. The question is it useful? So for those of you who are familiar with machine learning approaches, have seen before, you have like a validation strategy here. So this is what we did to convince ourselves that this is remotely useful or valid. So we took about 4,000 compounds from what we call the MOA box. This is published maybe 5 or 6 years ago out of Novartis. They're compounds with known targets. We know their mechanisms of action. And we have those profiled in a cell painting assay. This is just a standard fluorescence assay. We use images from the highest compound concentration only to ensure that we're getting diverse morphological changes. And we also have, for each of these compounds, a chemical fingerprint, and we have a pQSAR profile, so a sort of bioactivity fingerprint. And we trained two different models. One is we call our baseline diffusion model. So instead of having that prompt be a sentence, we're having that prompt be the chemical fingerprint. So it doesn't know anything about its biological activity beyond what's being presented in the image. It only knows about the compound structure. We also trained a pDIFF model where we give it that bioactivity. We want to show that there's differential performance gains by giving it that pQSAR fingerprint. And we validated this using what we call a realistic held out test set. So you can imagine that in some of these training sets, there may be analogs, chemical analogs. If you have two analogs, one's in the training set and one's in the test set, that's kind of cheating. You can guess that similar compounds will produce similar results. So instead, we cluster all the compounds based on chemical similarity, sample the largest clusters to build the training set, and validate with the singletons and the smaller clusters, the most chemically distinct. And the Tanimoto similarity is like 0.1 between those groups. So it's truly what we think it represents what happens in reality when you're working on a discovery project. And what we're looking at here first is just the correlation between the features that we get in a real image and a generated image, right? You can say it looks cool or it looks about right, but really, what does that mean? Well, it turns out it does look cool. If you look at a few different examples here, so this is done in U2OS cells. Halofuginone is an inhibitor of osteosarcoma. We can see in the real images, we're basically seeing cell death, and pDIFF recapitulates that. The baseline, based on chemical fingerprint only, just looks like a bunch of cells oddly stained. If we look at a PKC theta inhibitor, similar thing. Like the real image looks quite similar to pDIFF, and the baseline model doesn't perform so well, and it goes down the list, right? They don't always look the same, but they tend to look a lot more similar in pDIFF and real images. Of course, looking the same or similar is not real science. We want to get some data on this and quantify things. So it turns out we can do so with some basic imaging features. One being coverage. That's like what fraction of an image is covered by cells. One being cell count, simply count the cells in there. And also to look at the cell size. So we have real images too, and we want to compare the pDIFF generated image to the real images. That said, there's a lot of variability in the real images. So the first thing we did was sort of take an upper bound and split the real images into two groups and compare those to each other. And that establishes this upper bound. Like, what's the best we could hope to do if we were perfectly emulating real life? And so you can see that for the coverage, we have 0.67. These are Spearman correlations. The cell count, 0.56. Cell size, 0.48. Now, if we look at the images generated by the baseline diffusion model, there's almost no correlation across these features of coverage, cell count, and cell size. But for pDIFF, we're getting a lot closer. The coverage is almost as good. The cell count's about half as good, and the cell size is less good. Again, though, it's not perfect, but that's not what we're after here. The question is this useful in any way? So like we showed for the in-silico labeling, we've done a similar experiment here for virtual hit expansion. We want to retrieve molecules that induce a similar phenotypic outcome to known active molecules. So we took about 100 compounds from the training set and took their real images, and we first compared them to the held out real images, and we pulled a sort of fingerprint representing those images from them to do this comparison. And we want to compare our ability to retrieve nearest neighbors with real images versus real images and real images versus pDIFF images. And so our performance metric here is just simply the overlap. So take a molecule, what are the most similar real images, the top 50 most similar real images, and do the same thing with the generated images. Look at that performance overlap. So we have a number of different tests that we've run here. The first way we did it is instead of using 100 compounds and pulling their real images, we actually just do sort of a random test. If we take 50 random images, what's the likelihood that you capture some of the nearest neighbors that you do with the real images? We also do a test with the chemical fingerprints and the pQSAR fingerprints alone, so no actual imaging model, just taking those vector similarities. And then we also did the baseline diffusion model. So this is just a generated image based on the chemical structure, and the pDIFF model. So what we see here is actually, not surprisingly, the chemical fingerprint to chemical fingerprint does not retrieve similar images much better than random. That pQSAR alone, without the image process, does do a good job at retrieving similar compounds. That said, there is a wide spread here. There are compounds that completely fail, for which we're not able to do any phenotypic expansion. We have very low overlap. It does very well in some cases, but poorly in others. The baseline diffusion model does a little bit better than the fingerprints alone. There's something captured in doing the imaging, but when we do pDIFF, we're able to pull out basically half of the ground truth molecules on average, even up to 60%. And importantly, it's a much tighter distribution here. We're consistently getting around 40 to 60%. So this gave us enough confidence to begin applying this to projects. I wish I could show you those results. They're extremely exciting. I cannot. Maybe you'll hear about them in 5 years or something. But we do have our first project impact in the last 6 months. We're seeing about a 12-fold hit enrichment when we apply this in a phenotypic expansion context. And additional hit expansion is ongoing. And one really important thing here is that we can do this for any compound. We can generate an image from, say, a make-on-demand or ultra-large library, chemical library. It just costs us the computational time. We don't even have to order the compound. So we can order them after we've decided whether or not it's likely going to be a hit. Okay, so just summarizing real quick, and I'll give you some food for thought, and then I'll wrap. I hope I've convinced you that image analysis is the most mature application of AI in industry. I certainly feel that way. We're going on almost 15 years now of AI for imaging, which is a long time in this space. Also, I hope that I've convinced you that when you bring modern instrumentation and creative assay development, we can really quantify biology in unprecedented ways with these AI models. We can do things we've never been able to do before. And while I wouldn't say that we're ready to replace all drug discovery processes with AI, these generative approaches are showing some promise in accelerating discovery, and sometimes augmenting or sometimes even replacing portions of the experimental workflows. So what's next for imaging? I don't have any work to show on this, but one of the things we're thinking about is some of these new masked autoencoders. Basically, they allow you to get even more accurate, meaningful representations from images by training in a different way. So I showed in ISL, in the In-Silico Labeling pDIFF, this notion of image similarity is key. Well, we're doing it based on a certain fingerprint, but if we had an even richer one that understood what was going on in the image, that would be better. So last year, Recursion published a model that was adopted from the Facebook model that shows that it works super well in cellular biology. You can do things like cluster images, find new phenotypes, assign MOAs, do QC, detect anomalies. So I think this will be where we're heading next. But of course, imaging is not the only space where we're seeing AI disruption. There's an active revolution right now with these transformer models. There's new chips, and the number of different tasks that they're being applied is really quite amazing. This is a picture I took last year in Basel. When my Swiss German is not so good, but I can hold my phone over a page of text and it'll translate it on the fly. I'm sure folks have heard about AlphaFold, predicting protein structures from sequence alone, ChatGPT, and this revolution in these models are being applied to structure prediction, transcriptomics, generative chemistry, multimodal data integration, et cetera. And it's still scaling right now, and we're still looking for new applications. So with that, thank you so much for your attention, and I look forward to your questions.
Thank you so much, Peter. That was really, really amazing. I mean, yeah, just mind-blowing some of these developments that have happened. And yeah, like I think people probably didn't appreciate that we already used AI for 15 years in imaging. So that's also really interesting to know. And yeah, these applications, how they're getting more and more exciting and showing actual results in projects is really, really cool. So yeah, thank you for showing that. Maybe I'll start with the question. You mentioned that you're currently screening the entire Novartis library of 2 million compounds with brightfield microscopy, and then kind of predicting those images. What kind of time scale does that take? Like, how long really is that whole process?
I think the screen for brightfield for the full archive, and maybe someone in one of my collaborators will see this later and correct me, but I think it's about 6 months we project for brightfield.
Wow.
Now that we have the trained models, right? There's the downstream computation, and that's not trivial. Organizing the data, and there's all these sort of hidden costs and times. But I think overall, it should take us less than a year to go from the conception and start of the run to the point where we have these fingerprints in the database.
Yeah, that's you get such an amazing high value data set in really short amount of time. And then I was wondering for pDIFF, of course there are like different cell types you can stain. So like how do you decide like what cell type you would do the phenotypic screening with, and how many cell types at the same time do you do this with?
Yeah, that's a really good question. And I don't know if we have a good answer for that yet, but I can tell you that when we do run pDIFF, we need to build a model for that specific cell type. So it's not that we're able to come in with a larger foundation model and just do these expansions on any project. Rather, we need to work with the project team, have them generate some initial screening data and images that we can then use to train a bespoke model for that project. We've gotten the training times down to within a week now, which is great with using some newer computational methods. We're trying things like mixed cell types, more complex models. But I would say, yeah, we don't have a real recommendation or rules yet. We're still playing. This is pretty early.
Yeah, super cool. Yeah, another question here in the audience is, are there specific artifacts or errors that these image generators tend to create, and how easily is it, or how easy is it to pick a generated image from a real one?
Uh, so the first question around artifacts, I would say that um, the generated images, at least for in silico labeling, either tend to just work really well or it's total crap. Like it's very clear qualitatively that something has gone awry, and when you look at the pixel intensity correlations, it'll be very low. And you could just say, this one didn't work. So we have some cutoffs for like when we say like, okay, is the model working well enough just at the image generation level? To my untrained eye, I cannot tell the difference anymore between real microscopic images and in silico generated ones, unless there's something really wrong. There, some folks have said, oh, that could be real problematic for folks fabricating data. I agree. It could get a little crazy. But yeah, it's
Yeah.
really hard to tell right now. And you can see it in public life too on social media with the videos that are being created, right? You can
Yeah.
usually tell, but less and less so over time.
Yeah, yeah, that makes total sense. And when you were talking about this staining step, right, with the 10 different dyes potentially, how much time does that typically save? Like, how much time would it typically take to stain with those 10 different dyes? And I think you also mentioned a couple of disruptions that these dyes could cause, like they could be cytotoxic and you could disturb the image. So, can you maybe give us an idea of all the time that's being saved and maybe a few additional advantages of skipping this experimental step?
Hmm. Yeah, so I don't know the exact numbers here, but basically we see around an order of magnitude acceleration from doing brightfield alone versus doing multiple stains. And we have a time limit on how long after staining we can image. It's typically maybe within 24 hours. We can't even do like 2 days out for some of these stains. I don't know which ones they are or how toxic they are. I just know what the imaging folks tell me. Um, I would say overall, and this is something that we're trying to figure out, because there is a cost too of doing the bright field images up front. Like
Mhm.
let's say you want to do in silico labeling on a project. We encounter resistance from some project teams who are not doing a lot of screening. They're doing a few compounds and a few stains, and it's just not worth them adding on the bright field and generating those paired images that we need to do the initial training. Obviously, that cost is amortized by the amount of downstream screening that you're doing, right? In this case of like the entire Novartis deck, doing 50,000 compounds times 10 stains is totally worth it if you can get fingerprints for 2 million compounds. But that ratio, I don't know the exact time ratios, but it is something that comes into discussions just around like the practical considerations there. Some teams are like, it's not going to be faster, especially if we know there's no guarantee that the ISL model will work. Um, so I don't know. That's sort of a roundabout answer there, but
Yeah,
hopefully that gives you
amazing,
an
amazing.
idea.
Yeah, great. We have another question here in the chat as well. Uh, what are the compound concentrations you find most useful for mechanistic studies? You mentioned using high concentrations. At these concentrations, do compounds have off-target activities? So, yeah.
Yeah, that's a really great question. Um, so for the one test that we did at the highest concentration there for pDIFF, we really wanted to induce morphological changes because we're conditioning on these bioactivities, but if there's no morphological change, the model's not going to learn anything except what normal cells look like. In
Right.
general, though, we absolutely do see odd effects at those higher concentrations for some compounds. And we of course typically run at different concentrations. So it might be 10 to 12 micromolar, 1 to 1.2 micromolar, 100 nanomolar, for example. Um, that might be a typical set of concentrations that we'd run these experiments at. One of the cool things that I didn't get into is that we have interesting ways of incorporating more than one concentration into our analyses now. looking at those changes over time. So you may see no effect at a low concentration. You may see on-target effect at a medium concentration. And at a high concentration, you can see toxicity or polypharmacological effects. We definitely see that, and that can confound our analyses for sure. But I
Yeah,
think some of the newer models are able to take that into account, which is
amazing.
cool.
Amazing. How do you actually then train on, what type of phenotype constitutes which type of biological events? I mean, what are the kind of different types of phenotypes you're looking at? Are there like any specific biomarkers beyond number of cells, cell size and shape, anything like that?
Yeah, those are the typical ones. You'll see like elongation, you'll see cell death. Yeah, changes in shape are indications. It's those are the typical ones, but it also depends on the stains that you're putting in there. I would say for the pDIFF
Yeah.
case, we're using cell painting, which is sort of it's like a nuclear stain, a cytoplasmic stain, and a mitochondrial stain. But for some of the earlier examples I showed, where we're tracking organelles, there may be very specific stains where you get totally different phenotypes that you're reading out on the volume or looking at the ratio of surface accumulated ligand to internalized ligand, and that could be your readout. So there's a lot of different phenotypes we can detect, and we sort of have a different model for each of them.
Amazing. Yeah, super cool. Yeah, another question from the audience here is, do you need single cell imaging for the NN analysis? And could you comment on the resolution effects on the NN image analysis?
Uh, you don't need single cell. So we're actually just taking the fingerprint from the entire image, not even segmenting out single cells. We're just getting a sort of image representation. And I referred to those newer models, the masked autoencoders at the end that Recursion and Meta are looking at as a way to get a sort of embedding of an image. I don't understand the resolution question. Yeah, I'm sorry. Could you repeat
Uh,
it maybe?
that's okay. I think the question was, could you comment on the resolution effects on the NN image analysis?
Yeah, I'm not sure I understand the question.
That's okay.
Okay.
Yeah, another question is here. How well does this approach work when we look at SAR compounds in the lead opt? Sometimes even like subtle changes can have like a huge impact. So, I guess the question here is like, can this methodology be applied beyond the hit finding stage and even at a lead optimization stage?
Yeah, that's a really good question. Um, Part of that would be, well, first of all, we have not tried it in that stage yet, so I can't say for sure. Part of that would shake out in whether or not the pQSAR predicted activity changes. So if a small change between two analogs does beget a larger change in predicted bioactivity, it will definitely produce a different image and it would work. That said, if two analogs basically produce the same pQSAR uh, bioactivity profiles, you're not going to see a change. So it would really come down to that bioactivity prediction in pQSAR.
Yeah, makes sense. Cool. And yeah, maybe a few more questions here. Did you look at domain adaptation to cells that's more complicated to work with?
Uh, Domain apt. I'm sorry. I don't know what that means.
I'm not familiar with that either.
Okay.
So, all right. So maybe next question. What's the metric you use to know whether a compound is active based on the phenotype features?
hmm So in this case, we have a vector representation of the image, and we know what the DMSO images look like. So we sort
Mhm.
of have a distribution of what normal looks like. And then we say, is this vector representation of the image substantially different, um, 2 standard deviations, say, from DMSO? And then we call that, actually different.
Yeah, that makes sense. Cool, Peter. I think we had a few more questions here, but I think in the interest of time, let's wrap it up. want to give you a huge round of applause here. I mean, this was such an amazing talk. I mean, so inspiring. I think what you can do with these technologies, I think it's always great for people to see actual applications of AI and how AI is actually accelerating drug discovery programs. And I think imaging is really such a perfect example. And yeah, really thank you for illustrating this and really giving everybody hope and basically looking forward to your publications in 5 years when you can actually talk about those projects that this had an impact on. So thank you again, Peter, for an amazing talk, and thank you to our audience for dialing in today and for all the engaged questions.
Thanks so much for having me, Dennis. Appreciate you and all the folks at Drug Hunter.
Awesome. Yeah, thank you so much. And with that, we will be back on October 30th here with Dan Nomura. And we're going to be talking about reimagining druggability with chemoproteomics platforms. So I think Dan also had a collaboration going with the Novartis Emeryville side. And we're excited to hear more about some chemoproteomics platforms and how that could have a huge impact on the future of drugs as well. So thank you again, Peter, for your time, and wishing everybody a wonderful rest of their day.
Thank you.
Bye-bye.